Hi, everybody. Me again. And I'm going to explain to you the middle part of this last section of Chapter 13 that we're working on, which is gene expression. And hopefully at this point in time, you have already watched the Amoeba Sisters video over prokaryotic gene regulation. And now I want to go through eukaryotic gene regulation because it's a little bit different from prokaryotes. So just to recap, I know we review this a lot, but prokaryotes are single-cell organisms, and so they have not as much DNA, and therefore they don't have to have as complicated regulation sequences as eukaryotes do. Eukaryotic cells, if you remember, are usually a part of a multicellular organism. And multicellular organisms have a cell specialization. So we have a bunch of different types of cells that make us up. Muscle cells, blood cells, bone cells, neuron cells, we have a ton. And all those cells contain the same DNA molecule, but yet what makes them different from each other is the fact that each cell is only using some of the genes on that DNA molecule in order for them to um, complete the job that they're supposed to do for our bodies. So as we go through eukaryotic gene regulation, we're going to see that one of the differences between prokaryotic and eukaryotic gene regulation is the fact that unlike prokaryotes who turned on genes in groups, and if you remember, that group of genes that got turned on together was called an operon. Eukaryotic gene regulation, every single gene is turned on individually, not in groups. So it kind of reminds me of my classroom and the light switch. I don't know if you remember, but I have one light switch that turns on all the lights. That's kind of like prokaryotic gene regulation. Eukaryotic gene regulation would be if I had to have a separate light switch for every single light that is in the classroom. And that's what eukaryotic gene regulation is. Every single gene has to be turned on individually, or you'd have to turn on a light switch for every single light in the classroom. And because of that, there's a lot more DNA gene regulatory sequences that are involved in eukaryotic gene regulation. But again, the reason why we have to have that is because that allows for cell specialization. Muscle cells can then go on and just turn on the genes that they need to do their jobs. And a bone cell can go in and just turn in the genes that they need to do their job while keeping all the other genes off. So that's why it is a lot more complicated than prokaryotes. I'm going to skip this one and just kind of go to the diagram and talk through the diagram. So one of the things I want to call to your attention are the purple little circles, which here are identified as transcription factors. And as the name implies, transcription factors help the cell either perform transcription or they could even prevent the cell from doing transcription. In this case, we're seeing a diagram where it's showing how it's going to help with transcription. And again, if transcription is happening, that's when the cell is copying a segment of DNA into mRNA, and then the mRNA is going to go out into the cell, and the cell will start building the protein that that gene is encoding for. And so what these particular transcription factors are going to do is they bind to a region that can be pretty far away from the actual gene instructions called an enhancer. They will start binding to the enhancer, and they also bind to this DNA regulatory sequence called the TATA box. It's called the TATA box because it's a sequence of TATAA, TATAA over and over again. And so those transcription factors, as you can see here, they kind of bind a little bit to the enhancer. The ones at the bottom of the group bind at the TATA box, and that bends the DNA, but it also provides a site for RNA polymerase to come in and join up on the DNA molecule. And hopefully you remember, RNA polymerase is the main enzyme that helps in transcription. It's the one that will start copying the DNA into mRNA. So we need that for transcription. So in this case, these transcription factors are helping RNA polymerase come and attach to the DNA so that it can go down the DNA strand, and here's the gene. It's going to start transcribing that DNA into RNA, or turning that gene on, is what it's going to be doing. It's going to be expressing that gene and turning that gene on so that cell begins to do, or that cell begins to make that particular protein that it may need to accomplish that cell's specific job. So that's kind of the different parts that are needed to do eukaryotic gene regulation. Another thing that's kind of interesting that we want to talk through is a process that's fairly new that they call RNA interference. And so for me, it kind of reminds me of when I was little, and maybe I was playing Foursquare. You guys know what Foursquare is. And maybe you're playing Foursquare and out at recess, and all of a sudden another ball from another group of kids comes in to your game, and because of that, you didn't do a very good job. You might have gotten out. So you go ahead and yell interference, which means you want to do it over because something kind of interrupted. And RNA interference is kind of similar to that in that sometimes a cell has made a bunch of mRNA, and the mRNA is supposed to go out and make the protein. But for whatever reason, the cell decides, we don't need that protein anymore, or we already have enough of that protein. We don't want those instructions on the mRNA molecule to actually go out and get translated into a protein. We want to interfere, or we want to stop that message from actually getting carried out. And so scientists have discovered some RNA called microRNA. So we're talking little tiny RNAs because RNA is tiny to begin with, and these are microRNAs. And microRNAs are produced through a process called transcription. And even though they're a lot smaller than a normal mRNA molecule, they're going to become even smaller. So I'm going to go to the next slide that has the diagram of it here. And so you have this mRNA here that kind of folds into, they kind of call it a hairpin loop. So for us girls out there, it's kind of like a bobby pin, kind of folds in that direction. And then there's an enzyme that comes through, and this is fun because it's actually appropriately named. It's called the dicer enzyme, and it dices this mRNA into a bunch of tiny RNAs. And so now they're called MI, that stands for microRNAs. And then those little tiny pieces of microRNAs, they will attach to a protein called a silencing complex. And again, it's appropriately named because the job of a microRNA, once it has become a part of a silencing complex, is to go and attach itself to an mRNA molecule so we can interfere with the mRNA molecule so it can't go to the ribosome and can't produce the protein because we don't need the protein anymore. So this is our microRNA in the silencing complex, and how it attaches to the mRNA molecule that we no longer want to be translated into a protein is simply by base pairing. So this little microRNA will find the segment on the mRNA molecule whose bases are complementary to the ones on the microRNA, and they will eventually, attach. I'm coming. There we go. They'll eventually attach. And when they attach through base pairing, then it breaks the mRNA up. So it has here, they chop the mRNA up into little pieces. And now the mRNA molecule will not be able to do translation. And if there's no translation, there is no protein. The cell has successfully interfered with that mRNA message, and the cell will no longer make the protein. So that's kind of something cool. And if you research more about it, they're actually kind of doing some researches to whether we can make maybe medicines that have microRNAs that could possibly go in and stop people from making proteins that maybe they are producing because of a genetic defect that the body really doesn't need. That would be kind of exciting new ways of treating diseases if they can figure that out. All right, so the last thing I want to talk to you about is, again, the reason why gene regulation or expression in eukaryotes is so much more complicated than prokaryotes is because we need differentiation. We need all of our cells to have all the different cell types with those specific characteristics and specific jobs. And that's going to happen early on in embryonic development. So as the baby is developing in the mother, very early on after fertilization, differentiation is going to begin. and those cells are going to start being created. The different cell types are going to start being created, and hopefully in the correct spot. So the final video I have for you to view today is a video over something called Hox genes. And Hox genes are genes that get expressed early on during embryonic development that actually help the body know which cells to make where, so that all of the cells that are in our head get created in the right spots, and all the cells in our abdomen get created in the right spots. All the cells in our legs get created at the right spots. So that's the final video that I'm going to have you watch for today. Let me know if you have any questions, as this is kind of a challenging topic. I'm always available via email or during my office hours. So hopefully it's going well, and I will talk to you guys later. Bye.